Good morning, everyone. I think we'll make a start now. So I'm delighted to welcome you all to the 2024 Postgrad Seminar Series. And I would just like to begin by acknowledging the traditional owners of the land on which we gather today, who are the Wurundjeri people of the Kulin Nation, and pay my respect to elders past, present, and emerging. We recognize that First Nations people were the original scientists and that their contributions continue to inspire and inform medical and scientific research as we know it today. So today's seminar is titled Multivariate Analysis and Integration of Omics Data, delivered by Professor Kim-Anh Le Cao from the University of Melbourne, who develops computational methods, software, and tools for high-throughput biological data. Professor Le Cao has a mathematical engineering background and graduated with a PhD in statistics from the University of Toulouse, France. She then moved to Australia, first working as a biostatistician consultant in a bioinformatics platform, then as a research group leader at the Biomedical University of Queensland's Fraser Institute. Professor Le Cao has secured 3 consecutive fellowships from the NHMRC and received the Australian Academy of Sciences Moran Medal 2019 for her contributions to applied statistics. She was also selected for the International Homeward Bound Leadership Program for Women in STEM, which culminated with a trip to Antarctica in 2019, and the Superstars of STEM Women Program from Science Technology Australia. So just a reminder that attendees are invited to meet and chat with her over lunch after the talk in the Tapestry Lounge at 12 p.m. And with that, I'd like to welcome Professor Le Cao to the stage.
Thank you, Rihanna, for the introduction. It's my great pleasure to present or to introduce some of the topics I'm working on. And I start first by just giving you a brief overview of my lab. We have expertise in statistics and computational biology. It's a diverse team of statisticians, bioinformaticians, data analysts, and software developers. And we really focus on multidisciplinary research, developing software that are accessible for the community. And I show you today some of the methods that we have developed in my lab that are often technology agnostic, so they can be applied to any type of omics data. And I talk about my software, Omics. It's been selected for the Australian Yoiker Prize last year. So just to give you a bit of context, we have really changed the way we see biology today. And we really evolved from thinking about in a very reductionist way about biology, where we would think, well, this gene is having this impact on the system I'm looking at. Therefore, from a statistical perspective, you would run a statistical test to test whether that hypothesis is true. And now we're moving to a more holistic approach where we can measure hundreds of thousands of parameters. And I call them molecules in a broader way. So it can be a gene, it can be a protein, a metabolite, it can be a bacteria. But you can measure all of them at once. And so it's very difficult for a statistician to direct the analysis because there's no very clear question here. So it's hard to work out what might be the statistical test that is appropriate for those kind of data. And some of you have already realized that in this cutting-edge field, you really need to work very closely between biology and statistics, and bioinformaticians as well, of course. And this is because a lot of the statistical methods we want to use are very limited in this context, where you have a very small number of individuals and a very large number of parameters that you're measuring. And so a lot of the statistical methods that are the classical, traditional methods we use are very limited. So basically, you violate a lot of the assumptions from the statistical methods, and therefore you don't know if the results are valid or not. And so it's really important then to talk to the biologist and say, do my results make sense? Because I cannot validate it statistically. Of course, we have now a limited amount of data coming from multiple sources, a lot of computational issues to face. As I said, the interpretation is really critical for validation. And as you all know, we have new technologies coming every year. And so that creates also a lot of challenges for computational biologists. I'm going to talk about omics data and roughly any types of data that have been generated, usually using high throughput techniques. So from transcriptomics, so the measure of the expression of all transcripts, I will talk about proteomics, metabolomics, but I will also talk about the microbiome. And we also consider, so in my research, all those data sets I consider them as blocks of data, and I also consider clinical data as one block that I want to combine with all this information. And what is quite challenging, I think, for a statistician is that the relationship is not linear between all those layers of information, because we know that there's a lot of crosstalk between those omics data. So it's not like this line that goes, oh, from the transcripts you go to the proteins and so on, it kind of go all over the place. So I talked about a reductionist type of approach where we measure, for example, the expression of one gene across a cohort. What happened about 20 years ago now is we moved to the omics field where we measure the expressions of all the genes or all the transcripts across the cohort. And we would do that also for the proteomics and so on. And in the past 10 years or so, we've been really focusing on the multi-omics approaches, where you do the same. You measure all these omics, but they are measured on the same individuals. And basically, what we're trying to understand here are what are the genes that explain my biological system, what explain disease, for example. But I also want to understand how correlated are those genes. And I want to understand how correlated or associated are some genes with some proteins. So this is what a multi-omic approach is trying to do. But basically, from this analysis, we're trying to reduce the size of those data to something we can comprehend. Because eventually, we want to go back to a reductionist type of approach. We want to be able to validate those results. There are different types of methods for what I would call data integration, so integrating or combining all these different layers of information. The one that we will be focusing on today are matrix factorization techniques. And basically, I will show you how they help you to reduce the dimension of your data and have a better insights into the system you're looking at. But there are all other different types of approaches, from very basic methods that people still use today, where they will analyze one omic layer at a time. They may identify the key genes, the key proteins that explain, for example, disease or treatment effect. And then once they get a smaller subset of those genes and those proteins, they can have a look at the correlation between them, for example, and then make sense of it. So that's a very basic approach. You don't really, you integrate in the late stage of your analysis. Whereas the methods I will present today, the integration happens in the early stages. Right when you have the data, this is when you start to integrate. And I will talk about multivariate analysis because this is something I've been working on for the past 15 years now, where we're really trying to shift this one gene hypothesis paradigm by using those methods. So I will talk a lot about combinations of markers or combination of parameters that we measure, rather than univariate markers, which are usually identified with univariate statistical tests. So for example, if you do a t-test, or if you do an ANOVA test, you run it per gene. And so you completely ignore the interaction between different genes or the correlation or the association. I'll show you how these methods can be used for dimension reduction and how they can be extended then to integrate different layers of information. And I also mentioned that we can also do supervised analysis or classification using those methods. So I first start with one of the workhorse of multivariate analysis, which is principal component analysis, and then we're going to go through more advanced methods. So PCA, Principal Component Analysis, is, as I said, it's a workhorse for multivariate statistical analysis. I use it all the time when I have a big data set, even when it's small, because I want to understand first what is the quality of the data, what is the signal I can extract from it, what is the data structure, are there things that I didn't anticipate to get from the data? For example, is there any source of variation that are due to batch effects or experimental mishap or whatever? PCA is very good to tell you, to give you a snapshot of your data into something that is much better for us to interpret. So in PCA, what we do is we replace the original variables, so the parameters we measure, by new variables, and I'll show you how we do that. And those new variables are called principal components. And those principal components are supposed to summarize as much information as possible from your original data set, but in a much reduced dimensional space. And in PCA, this information is actually the variance that we get from the data. So I'll give you some example here about what a linear combination of variables is. So assume we've measured two variables, height and weight, in 10 individuals. But instead of looking at the height and the weight, I'm going to combine them. So you can take the average if you want, and then in that case, you would assign a coefficient that is 0.5 to each of them. Or you can do another combination of coefficients. So here, for example, I decided to apply the coefficient 0.5 to height and a coefficient of 2 to weight, because I say, well, actually, I think weight is more important than height for the problem I have to deal with. So you can calculate that. This is basic algebra, and you get a linear combination where for each individual, instead of having two values, you end up with only one because you've combined them with those coefficients. And so this linear combination is a component. So you can do that very efficiently now. We have algorithm in R and other languages to do that. But really the challenge is that how do you define what is the coefficient you assign? How do you decide that? I can show you another example that is a bit more that is larger. So here we have still 10 individuals, and this time I've measured 5 variables. If you plug that into a PCA, you're going to obtain something that looks very similar to this data set. You still have your 10 individuals, but this time you have those principal components. And I want to remind you that a principal component will be a linear combination of those 5 variables, so it looks different from the original data, and that they will replace completely your original data set. And those principal components are designed to explain as much information as possible. Because we're interested in dimension reduction, meaning that we don't want 5 variables, that's way too many. We want maybe 1 or 2 variables. And so we may only consider, for example, the first 2 components and say that's it. That's enough. It will explain as much information as I'm happy with from this data set, and therefore have reduced the dimension of my data from 5 to 2. So I talk about information and variance. If you calculate the covariance of your original data, this is what you get. Okay, so you have, on the diagonal, you have the variance of each variable. And outside the diagonal, you have the covariance, which is very similar to a measure of correlation. So covariance, so a correlation is between -1 and +1, it's bounded. Covariance is between -infinity and +infinity. That's the difference. So if I calculate the variance of my data set, basically what I do is I add up all the variances of all the variables, and I get a value of 3.27. That's the variance of my data. If I do the same, so if I go back to my PCA result here, and I calculate the covariance, if I add up all the variances on the diagonal, I get exactly the same value as before. That's why I'm saying we're trying to extract as much variance as possible, And then we say, oh, but actually, you know, the amount of variance explained in the later components is very small. So I'm not going to consider that. I think it's just noise. And so you may end up with principal component 1 and 2, and you will explain as much as you want, as much variance or information as you want. And the other thing that is interesting here is that the covariance values outside the diagonal are all zeros. And that's because the way we design those linear combination of variables is such that they don't explain the same thing. And so I come back to that concept of principal components are orthogonal, but basically they are, in order to extract as much information as we can, it's not useful for us to re-explain again and again the same information. Okay, so I talk about matrix decomposition technique. I told you that you could summarize a data set using those components, and basically that's what we're trying to do here. So you have your original data set here. You have the samples in rows and metabolites or genes in columns. I explained to you that you can extract several components and they will summarize the information from your data. And I also told you that in order to obtain those linear combination of variables, you need to know what are the coefficients assigned to each variables. And those are actually stored in something called a loading vector. So for each component, we have one loading vector, and each loading vector is going to tell us, well, the coefficient for variable 1 is this, the coefficient for variable 2 is this, and so on. And so, as I said, the hardest part is actually to find the loading vector. Once you have this, the rest, you know, you can calculate your combination and so on. And so this is done statistically. We optimize. Basically, we estimate those coefficients by asking to maximize the variance of each of those components. So this is done automatically in those methods. And one of the tricks of PCA is to decide how many components do I need. Do I need 2, 3, 10? So you probably, if some of you have used PCA before, you've seen that kind of graph. It's actually a bar plot where each component, each bar represents the amount of variance that is explained per component. So for example, this one explains about 60% of the total variance. And then the second one explains about, I don't know, 15% or so. And you can see it decreases. By construction, the amount of explained variance will decrease across the components. And so you can choose, for example, one component, or you can choose 2 or 3, and so on. So there's no clear decision-making, like there's no clear method to decide how many components you need. It's very arbitrary. So the first one is you say, well, you know, I look at this and it obviously decreases, so I don't need 5. From 5 to 10, I'm not going to explain anything. That's probably noise. But then do you include number 2, 3, and 4, for example? So one practical method is we use the elbow method. So basically, when it kind of sharpens and flattens is where we should stop. The other one is also to look at the proportion of explained variance. So you add them up. So for example, if you use one principal component analysis, one principal component, you explain about 60% of the variance of the original data. You could say, well, that's enough for me because I know my data set's very noisy anyway. If you include the second component, then you add up what you've explained on the first component and the second component, and you get about 0.75. And you may say, okay, that's enough. So either you use the elbow method, and in that case, I would use only one component, or you can say, well, actually, with one component, the visualization is going to be very hard because we're going to have 1D visualization, so maybe you want 2. So there's a bit of, soft touch on how you choose those components. But just remember that PCA is an explorative method. So you're not inferring anything from a PCA output. You're just saying, this is what I can see from the data. These are the sources of variation I can interpret from this data. There are statistical tests who actually just help you to decide the number of components, but I wouldn't advise you use them because they have a lot of, they're very limited for big data. Okay, so you can look at so this is a PCA sample plot of a very, a toy example, where here the samples that were measured, so in the rows, are different type of fruit. And then the variables or the parameters that we measured are different characteristics of the fruit, for example, the texture, the acidity, and so on. So if you take that data set and you plug it into PCA, you would obtain a plot like this. It's called a sample plot. So the fruit are my samples. And basically, how we obtain this plot is we plot component 1 versus component 2. So just remember, before you had say we have 1,000 characteristics of this fruit. We reduced it down to only 2 components. And then we just plot those 2 components. And it can be seen as projecting your data into a much smaller space, which is a space of only created by those 2 components. But remember that we have each of those components are linear combination of all those characteristics. So we still retain that information. And often, depending on your data, often those components have some sort of meaning. So for example, here, the major source of variation that has been extracted from those components is how easy or difficult a fruit is to eat. So that would be the meaning of the first component, so the horizontal, the x-axis. And the second meaning of the component would be how tasty and untasty it is for humans. So usually they have a meaning like that. And so you can see, for example, all these fruit are actually easy and tasty, and all these fruit are difficult and not tasty, for example. So this is how you can start to interpret your sample plot. The other plot that people don't know much about, but I think it's useful to mention, is how you represent the variables. Because at the moment, I only show you the samples, I show you the principal components, but what actually happens inside the PCA so that we understand better what is the correlation between the variables. And so for that, we use a plot called correlation circle plot, where basically we calculate the correlation between each variable and each principal component. So for example, here I calculate the correlation between my variable xj and the first component, and I do the same for the second component, and then I plot it. Because it's a correlation, my circle will be of radius 1, and basically it ends up here in the plot. Okay, so that's what we do. But what is interesting is if you do that for all the variables you have. So here I'd only show you 4 of them. And basically it's going to tell me that x1 and y1 are highly correlated together. So basically I look at the angle between those 2 arrows. And that angle, if I take the cosine value of that angle, it's going to tell me how correlated they are. So I can start to say, oh, okay, x1 and y1, those 2 variables are highly positively correlated together. If you have a flat angle like this, it means that x1 and x2 are negatively correlated together, but they're strongly negatively correlated together. And then if you have a right angle, like the y1 and y2, it means that they are not correlated together. So when you have variables that are not correlated together, it means that y1 contributes heavily to the definition of principal component 1, the x-axis, but y1 doesn't contribute at all to the definition of principal component 2, whereas y2 is the inverse. It contributes to component 2, but it doesn't contribute to component 1. So if you see their coordinate, basically tell us that. So you can start to understand the correlation structure of your data set. And that's very relevant in biology because we know that a lot of genes are correlated, and we know also that a lot of genes are not correlated. And how do we visualize that into a smaller dimensional space? When you have a lot of variables like this, we remove the arrow. So you will just see the name, often you will see the name of the variable. So you kind of have to do intellectually, mentally, you have to draw those arrows and kind of work out roughly the angle between them. And another plot that is probably not well known is the biplot. And basically it combines a sample plot and the correlation circle plot that I showed you before. So the sample plot, as I told you, tell us more about the samples, how similar they are together, what is the source of variation that explain why they're different. The correlation circle plot is going to tell us, how correlated the variables are in that reduced space. And the biplot is going to overlay those two plots. So you will have at the back, you have all your samples. And in the front, in red, you have your variables. So for example, here, you have a lot of melanoma samples. And you have a set of multi-drug transporters or drugs that are highly correlated together, so that's why they're clump, but they're also pointing towards the melanoma samples. So that's going to tell you that potentially those transporters may have a high expression in melanoma and a low expression in the other samples. So this is the kind of interpretation you need to work on. You need to, use additional visualization in order to get that, but that's what the PCA can tell you as well. Okay, so this was, an introduction to PCA and telling you a little bit more about what's happening in PCA. Also going through some less well-known visualization techniques, such as the correlation circle plot and the biplot. Now I'm going to move on to other types of multivariate techniques. They build on the similar concepts. It's just that we are going to add a bit more complexity in the analysis that we're doing. So the first one I want to go through is PLS discriminant analysis. It's used for classification and variable selection. So classification analysis is basically using a classifier or a supervised method to understand what is the relationship between the data and an outcome that you've measured. So it's very different from PCA. In PCA, I showed you, you just plug in your data set and you don't say anything about, oh, my patients are sick or not sick. Here you do. Here you say, well, actually, I'm interested in discriminating cases and controls. You can have different aims for classification analysis. You can you may want to just have a descriptive aim. So you want to, again, build those linear combination of variables, but so that those components discriminates your groups of samples. Or you may also want to have a predictive aim. So you have trained your model, and then you have a new data set, and you want to use exactly the same rules. You want to use exactly the same linear combinations to work out whether this individual is a case or is a control, for example. So it's more prediction-based. And of course, you can build those classifiers based on all the variables you have, so all genes, or you want to do variable selection, because you say, well, not all genes is going to tell me what's happening in my biological system, so I really want to only identify a subset of those genes. So I will refer to either a biomarker panel or a molecular signature when we talk about variable selection. And what is interesting with multivariate methods is that the diversity of your signature is quite high compared to univariate methods. So I'll show you an example here. We have each box plot represents the abundance of metabolites. So I only show you 9 metabolites here. On the x-axis, you have 4 groups of samples. Here, I mean, this is a metabolite data in yeast, but it doesn't really matter. It's different conditions. And this is a signature that was identified by a multivariate method. And so you can see the diversity of the profiles. Some metabolites are very good at discriminating a particular group or two groups at a time against some other groups and so on. If you had used a univariate test, so for example in ANOVA, you would only identify metabolites that look like this because you're just trying to maximize as much as possible the difference between one group and any of the other group. And so if that difference is very strong, this is the kind of profile you will always get as the strongest, the most differentially expressed metabolite. So multivariate methods are different because I told you they combine information from different markers. And so they try to identify a complementary signature. So you've seen that graph before for PCA. And this is what we do when we want to also do the discriminant analysis. So we still have our data set as an input, but I also add another input, which is the outcome. So the outcome is going to tell me, well, patient 1 has treatment 1, patient 2 has treatment 2, patient 3 has treatment 2, and so on. And this is what I want to discriminate. I want to discriminate the different treatments that those individuals have. I then do this matrix factorization technique that I explained to you before with PCA, where we extract the components and the loading vectors, except that this time we want to maximize the covariance between this data set and this outcome. So we add in the method, compared to PCA, we add an extra layer of information that says, can you give me components that combine all the genes that I have measured, but also so that they discriminate the different treatment groups. And then we can also do feature selection. So using the loading vectors, I told you there are coefficients that are assigned to each gene. Some coefficients are very low, they're close to 0. So basically it means that they're not very useful to define your component. And so using lasso penalization, so it's a technique that we use in regression, you can completely shrink these coefficients to 0, so that in the end you only obtain a loading vector that has only very high value and zeros. And the high values of those coefficients will correspond only to the genes that participate in the discrimination of your sample groups. So this is how you do feature selection. And then you can compare a PCA plot on the left and a discriminant analysis plot on the right. As I said before, PCA is unsupervised, so I didn't color my dots. So these are sample plots of 63 samples. And this is a data set with about 3,000 genes. In PCA, I didn't color my dots, so they're all blue, because I want to emphasize to you that it's an unsupervised method, so we don't know anything about the groups. Whereas in discriminant analysis, where I use PLSDA, I explicitly asked the method to discriminate my 4 groups of patients. Here they have different tumor subtypes. And so you can start to see some discrimination happening. So it already tells you, yep, I can discriminate my sample groups. Whereas in PCA, all you can say there are clusters of patients, but you don't know what it corresponds to so far. But you can identify outliers, you can identify, specific batch effects, and so on from there. Okay, so we covered PCA, data exploration, we covered discriminant analysis, which is a supervised analysis, where basically we use the same concept as PCA, but this time we ask the method, can you discriminate sample groups? Can you identify the discriminative genes in the linear combination that will tell me what's going on in this data set? So you could stop here. You can already, from a PLSDA plot, you can have exactly the same plot as the PCA. So you can stop here. You have a list of genes. You know how your samples are looking like in a reduced sample plot. And you can start your validation if you want. You can do cross-validation and all these things to validate your model. And then you could, if you wanted to, go back to the lab and test this signature, for example. But if you are interested in integrating different layers of information, then of course, you can go further. And so this is PLS. So PLS stands for projection to latent structure. And basically, it's the same concept as before, except that now we have two data sets. So for example, you have gene expression, and here I have metabolites. And I will also do matrix decomposition. So I will decompose this data set into components and loading vectors. And I will also decompose this matrix into components and loading vectors. Okay, so exactly the same that we did for PCA, except that this time I would say, okay, I'm happy to have those components, but they need to be correlated together because I'm trying to integrate this information. I'm trying to extract common information between those two data sets that are measured on the same patients, but they're actually measuring very different things. One is measuring genes, one is measuring metabolites. And so basically what a PLS model is going to do is it's going to try to find the optimal linear combination of genes that is going to be maximally correlated with a linear combination of metabolites. And so this is how we extract common information from those two data sets. And you can also do feature selection. So we're not going to take all the genes. We may select in the method only 10 genes or 50 genes. We may select 10 metabolites or 50 metabolites to build those components. And then you can complexify this for more than 2 omics data sets. So here I will just show you 3. But you can also do a supervised analysis, so you can also include the outcome. And so things start to get complicated because you start to extract correlated patterns between all these different omics layers, as well as integrating the outcome, so also wanting to discriminate your sample groups based on this integration. So this is one of the latest methods we developed, which is quite popular now in the field because there are not many methods that can integrate omics data sets that are also very visual. But you will see there are some other related methods that also use matrix factorization, matrix decomposition techniques to do the same. It's just that some of them may not do supervised analysis, or they have just different characteristics, I would say. So when you do this, you obtain a set of components per data set, and you know that those components should be correlated together because that's what we asked the method. So you can extract those components and you can plot them one by one. So that's what I do here. I have 4 data sets that I'm integrating. This is a sample plot, and I'm plotting component 1 and component 2 from the first data set, and I do component 1, component 2 for the second data set, and so on. And then I colored my dots according to different subtypes of breast cancer. And you can see that there is some consensus between the data sets. We can extract similar information, but with different quality. For example, here, the mRNA data set is very good at discriminating the basal samples versus the others, not so much in the microRNA, and not so much in the proteins. And so this kind of just visual output tells you a bit more about the quality of the ability of the method to integrate all this information into something that is common and into something that can also discriminate the breast cancer subtypes. So at that stage, you may say, well, actually, no, I don't want to include this data set in my analysis because I think it's going to be too noisy. I think it doesn't have the similar information to the other data sets, for example. So one mistake of people who want to do data integration, omics data integration, is they want to go straight to the integration. And it's important, I think, for me to go through PCA and PLSDA and all these things because each of those steps will really help you to guide your analysis further rather than jumping straight into this and then say, and then what do I do now? So it's really important when you work in that field to, take all these baby steps along to really understand the sources of variation in your data, the quality of the signal in your data set as well. And then I showed you correlation circle plots before for PCA, but we created other types of visualization because things get a bit more complicated when you deal with all these different data sets. But when you are still interested in the correlation or the associations between those variables. So we developed a circus plot where here on the quadrants you have the variables that have been selected by the Diablo method. And then, so each of them here is one variable. And then we have links between them if the correlation is either positive or negative. This is above a correlation cutoff of 0.7. So it can tell you just visually, what is happening. For example, here I can tell you that not a lot of proteins are actually correlated with any other microRNA or mRNA. And you can do the same with, a heat map, where you basically also represent those correlation or those associations. And the other thing I wanted to talk about is that we don't have a lot of p-values in multivariate analysis in general. One reason is that you can get a p-value that's going to tell you, yes, your linear combination is far from random, far from a random result where you randomly combine your genes. But that's it, because when you work in multivariate analysis, you have to consider variables as a set. And so, yes, you can get one p-value, it's going to say, yep, you're doing great. It's not going to tell you what each gene is doing. Because as soon as you want to know what is each gene doing, then you have to use a univariate test where you have a p-value per gene. So that's why you don't, the p-values are going to be less meaningful in multivariate analysis. Okay, I want to finish by just telling you a little bit about Mixomics, and then I give you an example of Omics data integration. We covered today PCA and PLSDA and Diablo, but there are some other methods we developed. I also covered PLS. But basically, Mixomics is our toolkit for multivariate data analysis, integration, and exploration. And what I tried to explain to you before is that they're all component-based methods. So once you understand how you can decompose your data matrix into a set of components and a set of loading vectors, then for us as our software developer, we can extract those components, we can extract the loading vectors, and we have very similar outputs for any of those methods because they all build on the same principles. And so we use the components, as I showed you, to represent the samples and how similar they are based on their profiles. And then we use the loading vectors to tell you more about how correlated the variables are, how important they are in your signature, and so on. So we developed 13 new methods, and we have a website, and so you can have a look at that if you're interested. But what I want to emphasize here is that every single method is designed to answer a very specific question. So in my slide, you saw that for PCA, I say, what is the major source of variation in my data? For PLSDA, I say, can I discriminate my sample groups? For PLS, it's, can I extract common information between two data sets, and so on. So you kind of need to decide first, what is your question? What is the question you're asking to your data? In order then to decide what is the analysis you want to do. Okay, I'm just going to go through one example here. It's a study that we did with people from the US and Canada that looked at the first week of human life. So here what those researchers did is that they managed to extract a very small amount of blood in newborns at different days during the first week of their life. So at day 0, 1, 3, and 7. And then they were able to do the transcriptomics, the proteomics, the metabolomics, and some other things. So we started with this data set here, which we call the training data set. It's a cohort of newborns from Gambia. We submitted the paper, and then they said there's no validation there. So we had to go back, and then we got a validation cohort. This is really what happens in research, right? Like what you say in the paper is not what you had planned. But they say, basically, you need to validate your results in another cohort. And so we went to PNG and got another cohort of 30 newborns. So what's the single omics approach? Actually, I mean, was insightful, of course, but brought way too much information for us. So this is a plot showing you the number of transcripts that are differentially expressed between day 0 and any other day. So for example, and whether they are up or down regulated. And so you can see across time that the number of transcripts that is differentially expressed compared to birth is increasing. And sometimes it's increasing a lot. So here, for example, we have 1,300 genes that are downregulated. So you can do that for every data set. And then a natural analysis to do is to say, well, I wonder if those genes are correlated with those proteins and those metabolites, and let's represent them into a network. And this is where it gets really messy, because you can't actually extract any correlation structure that makes sense to us. It's just everything's kind of correlated. So this is basically this approach. The single omics approach is more like a single omic layer analysis. And basically this is what they did. So analyze each omic independently, identify the key genes and so on, and then calculate a Pearson correlation between those variables, and then this is a network that I showed you before. If you use multivariate methods like Diablo, as I showed you before, first you have a bit more outputs, but also the correlation structure between all these different omics layers is much clearer. Because it's designed for that. It is designed to extract correlated information between data sets, and it will show you so. And so we found that there were very specific pathways that were identified in component 1 of the method or component 2 and so on. So we could confirm the analysis or the results we got from the single omics analysis using an integration method. But we could also find new things that we couldn't find before because it's a very subtle signal. And that subtle signal can only be identified if it's amplified by the other ones around, if I can say. So that's why you can also, through data integration, you can also identify this subtle signal because it's going through combination of variables and how those variables are correlated with other variables. So if you want to start with Mixomics, well, it's in Bioconductor. We have a lot of tutorials, vignettes on our website. We're doing an introduction workshop of 3 hours in a few weeks. We also do online workshops, so you can have a look at that if you're interested. And just to summarize, I really wanted to focus, the talk is not about Mixomics, but more about multivariate matrix decomposition. I wanted to show you that if you use PCA, it will tell you a lot about the source of variation in the data. If you use, those methods are also have been designed to do classification, and that starts to be quite interesting for us because you can do prediction, you can select variables, and so on. I show you that I think one of the advantages of these methods is all this visualization you can have around it. Makes it much easier for a biologist to understand also what's going on in the method. In general, the omics field, when you want to do data integration, is to generate new hypotheses. Okay, you're not actually trying to, validate a specific hypothesis. You're actually trying to generate it so that later on you can validate them. And I also want to say that both types of approaches, univariate and multivariate, are complementary. Some people start in their paper, they start by doing the univariate analysis first because it tells them already about what are the genes that are differentially expressed and so on. And then they move on to the multivariate approaches because they can't integrate this information if they have different omics data sets, or they can't visualize what's happening in their results. And some people do the inverse. They start with multivariate analysis first because they want to do all the data exploration, the visualization, generating new hypotheses. And then they go to univariate analysis on very specific markers where they will do very standard statistical tests. That's it. Thank you.
So thank you very much to Professor Le Cao for a wonderful talk. So we now have some time for questions, either in person or online. And we'll be handing out chocolates as well.
Thank you for the amazing talk. I just had a question regarding the discriminant analysis, and when you're choosing these groups, like at what point do you need to worry about overfitting your model? Like if I have 100 samples and 100 groups, I'm just going to be kicked out at validation.
Yeah, it's a fair concern. You should be worried all the time about this. So of course, when your sample size is very small, you will overfit.
Yeah.
And so this is why I tell researchers, if your sample size is very small, it's about how you interpret your results. If you say this gene might be discriminating those sample groups based on the data that I've analyzed, it's fine. But if you say this gene is the one that will, always save, life of people, then that's wrong. We do a lot of cross-validation in order to estimate what is the likely generalizability of those methods. So you should always use cross-validation. We do repeated cross-validation to make sure that it's not, some sort of luck result. And what I didn't talk about for the example with the first week of life study is that the validation set we use actually to validate. So we had build our classifier, and then we use the validation set to say, based on this signature and my classifier, can we predict which day we sample that newborn? So yeah, so in the best world, you'd have different data sets where you can validate those results. But a lot of time we don't have enough money to do that. And so the worst world is basically saying, look, that's what I see from the data, but that's not necessarily what's going to happen in other data sets.
Yeah, okay, thank you.
So we have a question from online, which asks whether you can say a little about like the computational demands of some of the techniques. For example, like the interactive times, or whether it's overnight, and whether you need like a standard desktop computer or a high performance one with GPUs.
So those kind of methods are actually very efficient to run on a laptop. So usually what I ask, I'm just going to pull out this one. I often say to people, you probably don't need to include your 100,000 peaks, methylation peaks in the integration. A lot of the peaks will be very noisy. So I ask people if they can only pre-filter the top highly variable features. That's what we do also in single cell. So that in the end, you may have up to 10,000 features per data set. And let's assume you have 5 data sets that can run on a computer. We've tried up to 15 data sets, it's fine, can run. What is becoming quite burdensome is when you try to estimate the performance of the method, because then you start to do cross-validation. It's repeated and it kind of starts to churn. And this is where you need high-performance computing. But it's mostly to assess the performance of the methods rather than to actually run the method. So if it's your final run, it will run fast. So often what I say to people is get a first pass first. You know, decide to select 5 genes and 5 proteins and whatever, and let's have a look at your results if they make sense to you or not. If they do make sense, then yes, it is worthwhile to do all the tuning, and the tuning involves how many components should I use, how many variables should I select per data set, and so on. So there's a lot of stuff, decision making. But I find often for, depending on the type of study, if it's a biological study, I often find that you can decide on those parameters almost arbitrarily once you understand your data really well. If it is a computational study, then yes, it makes sense that you need to do all this very fine tuning about those parameters. So yeah, quick answer is that if you want a first pass, it's easy to run. As soon as you start to be very specific about how fine-tuned your model should be, then you need high-performance computing.
Hi, thank you so much for the talk. So you were talking about when we use the elbow method. Do you have like a rule of thumb number on that for genomics data that you've seen?
You mean for the
for when you stop? Like choosing the number of PCAs, is it like, no, if it's explaining 80%, is that good enough usually?
No,
or is
no,
it like
because
99?
No, you can't do this because depending on the type of data you have, like there's no magic value. If you look at genomic data, usually the first few components don't explain much. It still decreases, but it doesn't explain, it may explain like 20% of the variance. And so it depends on your use. For example, for genomics data, you tend to include a lot of PCs because you use the PCs in a linear model later on. So it doesn't matter if you have 15, it's okay. I would say if your question is about what is the major source of variation in your data, that would be in the first few components anyway. So no, there's no clear guidelines. It's usually, and it's still exploratory. I mean, it starts to be very unwindly when you include 5 components because then you have to plot them like at most 3 at a time. If you have 3, we have 3D plots as well, but it starts to be very complicated. Yeah. So it's very data-driven process.
Thank you for your presentation. I just had a philosophical question that your experience might be able to answer. Your circus plot showed very little proteomic mass spec data integration with RNA-seq, and this seems to be a common theme. So my question is around, has biology been sort of corrupted and influenced by RNA-seq technologies, and the biology actually lies in proteins which have a function? And so when we look at breast cancer, we discriminate based on RNA-seq data, whereas proteins have different functions, and so there's a different grouping of samples. Is this a theme recurring when you do mass spec protein interactions with RNA-seq? And do you think that we're going to shift into looking at the proteomics and that post-transcriptional variance of integrations and exon inclusions in the future?
Yeah, it's a concern I've had for a very long time because, I talk to biologists and they say this should be correlated, and I say it is not. And I think it's a combination of both. It's the technology. Mass spec's extremely noisy. So I feel like the technology is still not up to scratch the resolution is not high enough for us compared to RNA-seq. It's a combination also of what we think is happening, but may not be happening, and anything in between. So other layers that also have this impact on the association between a protein and a gene that we still haven't measured. So yeah. So at the moment, those methods are data-driven, and they already tell us that there's not much in there, but we still don't know why. But it's very, I have it all the time. Like for me, it's very, very common.
Okay, I think that's all the time we have for questions today, but if you want to chat more with her, feel free to join her for lunch at the Tapestry Lounge. I invite you all to thank Professor Le Cao once again for the excellent talk today.